All right, let's gear up for a safari, but not the kind you might be thinking of. We're about to journey into a hidden world that is literally all around us, on us, and even inside of us. It's a world filled with these incredible creatures, totally invisible to the naked eye. We're diving into the fascinating world of bacteria.
You know, it's kind of wild when you think about it. Microbes like these, they make up at least half of all living matter on Earth. I mean, they're practically everywhere, in every single habitat you can imagine. So I think it's about time we get to know them a little better, don't you?
So, our guide on this expedition is the science of microbiology. This is the field that lets us understand these tiny, tiny life forms, from their DNA all the way to how they act. And their influence is just huge. They affect everything from the soil under our feet to our very own bodies.
Now, every good safari needs a map, right? So for our journey, we're going to start with the anatomy of a single bacterium. If we can understand its basic structure, we'll have our guide to this incredible, tiny world.
Okay, so here it is, our blueprint. We can basically break it down into three key parts. First, we'll look at the tools on the outside that bacteria use to, interact with everything around them. Then, we'll check out the protective armor that surrounds them. And finally, we'll peek inside at the internal machinery that keeps the whole operation running.
So let's start our exploration on the outside, shall we? We're going to take a look at the absolutely amazing toolkit bacteria have evolved for survival. I'm talking tools for movement, for sticking to stuff, and even for communicating with each other.
First question on the table, movement. I mean, in a world that's mostly liquid, being able to get around is pretty crucial for finding food or, getting away from danger. So how on earth do these microscopic creatures navigate their world? Well, meet the flagellum. And this is not just some tail that wiggles back and forth. No, this is a seriously sophisticated 360-degree rotating propeller. Seriously, think of it like a tiny outboard motor that pushes the bacterium through its liquid world with amazing efficiency.
The engineering behind this thing is just wow. You've got this long filament, which is made of a protein called flagellin, and it's attached to a flexible hook. That hook is then anchored into the cell by something called a basal body, which is the actual motor. All these parts work together to create this spinning propeller that is, and I'm not exaggerating, a true masterpiece of natural engineering.
Okay, so bacteria are great at zipping around. But what happens when they find a place they actually want to stay? How do they manage to stick to surfaces, especially something as slippery as, say, the cells inside our own bodies? They use these things called fimbriae. You can pretty much think of them as tiny microscopic grappling hooks that cover their entire surface. They're not for swimming, they're all about latching on. This is actually how pathogens like E. coli are able to get a foothold in our gut, which is, of course, the first step to an infection. All right, so they can move and they can stick. But get this, bacteria are actually kind of social. They can communicate and even share information with each other. I mean, how is that even possible for such simple, tiny organisms? The answer is the pilus. It's a special little hollow tube that acts like a bridge connecting two bacteria. Through a process they call conjugation, one bacterium can literally pass a piece of its DNA over to another. And this is how they share genetic secrets, including one of the most dangerous ones, antibiotic resistance. Okay, now let's move on to that protective armor we talked about, because it turns out not all bacteria are built the same way. There's this really clever staining technique from way back in the 19th century that helps us divide them into two major groups, all based on what their cell walls look like. And this is it, the Gram stain, developed by a guy named Hans Christian Gram. Here's the rundown. You start with clear bacteria cells. First, you hit them with a deep purple dye. Then, you use iodine to kind of lock that dye in place. After that, you give them a wash with alcohol. And for the final step, you add a red counterstain. And the color the bacteria end up with? It all depends on their cell wall. And here are the results. Bacteria that hold on to that purple dye, they're called gram-positive. The ones that lose the purple and pick up the red color instead, those are gram-negative. But why? What's the reason for this big difference? Well, it all comes down to their cell walls. Gram-positive bacteria have this really thick wall made of a mesh-like substance called peptidoglycan. It basically acts like a sponge, just soaking up and trapping that purple dye. Gram-negative bacteria, on the other hand, have a much thinner peptidoglycan layer, and they have an extra outer membrane. That extra layer stops the purple dye from getting trapped, but it lets the red dye stain the cell. And believe me, this one difference is a total game changer in microbiology. But, in the world of biology, there are always rebels. There are some bacteria with cell walls that are so different, they just don't play by the usual Gram-Stain rules. These are the specialists, the absolute masters of survival in extreme conditions. First up, we've got the acid-fast bacteria, like Mycobacterium. Their cell walls are coated in this waxy stuff called mycolic acid. You can think of it like a waterproof jacket that just repels most dyes. But if you use heat to force a special dye inside, it gets trapped and won't wash out, not even with acid. This very unique property is a huge clue for identifying the bacteria that cause diseases like tuberculosis and leprosy. And then you have the ultimate rule breakers, mycoplasmas. These guys don't have a cell wall at all. So what keeps them from just popping? Well, their cell membrane is reinforced with these molecules called sterols, which basically act like tiny support beams, giving them just enough strength to survive.
And finally, we get to the ultimate survivalists, endospores. When life gets really tough, some bacteria can form these dormant structures that are just unbelievably resilient. I mean, they can survive being boiled, frozen, zapped with radiation, and even doused in harsh chemicals. They can just lie in wait, sometimes for centuries, just waiting to spring back to life when conditions get better. It's incredible.
So, we've looked at their tools, their armor, their insane survival skills. But what's the point? Why does all this stuff actually matter in the real world? Well, it turns out these exact characteristics are the clues we use to identify bacteria and understand the diseases they cause.
This table really brings it all home. For instance, if a doctor knows that streptococcus is a gram-positive sphere, or what we call a coccus, they're a big step closer to diagnosing something like strep throat. In the same way, seeing a gram-negative coccus like Neisseria could point toward meningitis, and spotting an acid-fast rod, that's a major red flag for tuberculosis. A bacterium's structure is basically its fingerprint.
Our little safari has given us just a tiny glimpse into a world of absolutely breathtaking complexity, diversity, and resilience. It's a world that exists on a scale we can't barely even wrap our heads around, and it has such a profound impact on our own lives. This world was here long before us, and it'll probably be here long after we're gone. It really makes you stop and wonder, who's really in charge here?